I am Kerri Swanson. I'm the president of the board of Susan G. Komen here in Orange County, and I could not be more excited for today. What a beautiful day we have. Not only is it absolutely gorgeous outside, but the thought of this conference finally coming together and such a need being met by us all coming together is just, honestly, it's what got me out of bed this morning, and that takes a lot, let me tell you, on Saturday mornings. Maybe some people can sympathize with me in that space. So what a great day to bring together our family of women, men, and families living with metastatic breast cancer. And thank you for all of your efforts in bringing this conference together today. I've been involved with Komen for 20 years right now, after my mom was diagnosed with breast cancer in 1999 back in the Midwest. So tons of involvement with Komen. I can't think of a better way to be involved, and especially now that we are speaking to and bringing together our families living with metastatic disease. So we conducted focus groups, evaluations at educational forums, and talked to many women living with metastatic disease in Orange County, and we all agreed it is time to build a stronger community of support in our backyard, or might I say pastime. It's about time we did this. Today's conference program mirrors the findings of what this Orange County metastatic breast cancer community asked for. First of all, up-to-date, cutting-edge research and treatment information about clinical trial opportunities. We got a lot of work to do, and your involvement in that is critical. To learn ways to live their best, healthiest life via nutrition, mindfulness, and other integrative and complementary medicine modalities. Big words, but we'll learn more about what they mean today. To have a forum to create a united, unique voice to advance their agenda within the research and medical systems. But ultimately, we hope you use this conference to meet others who understand what you've been through and what you may face, connect, share experiences, take advantage of the resources provided, and continue to engage with us here with Komen Orange County. This conference is kicking off our commitment to have a future metastatic programming throughout the year. In your conference folders, you're going to find something very important. There are green evaluation forms. These forms, yes, they take a little bit of time to fill out, but they're not that much time. But your time is an investment in us providing programming that's meaningful to you. Without your input, we can't do that. So please, please take time to do those. And I'm forgetting where okay. Your feedback's important and as we build that programming. So a little bit of logistics. I hope I do this right. We are in the Pacific Room C and D. Somewhere there's a divider, but C and D. This is kind of our main spot today. Next door and across the hall, you'll see the breakout sessions. There's signage up high that kind of points you to where they are. There's Pacific A and B and some other breakout rooms over here. So when you get into that point of the agenda where you get to go and pick your topic, that's where you'll go. The expo you saw as you came in, and the restrooms are over here on the left. Okay, and before we get into some of our great, wonderful speakers this morning, I cannot say another word without acknowledging the incredibly hard work that a very passionate group of volunteers put into bringing this conference together. They donated their time and expertise over the last year. If you were on the Metastatic Conference Planning Committee, please, if you're able, stand, and we'd like to recognize you. If you see a committee member with a black indicator on their badge, please thank them, ask them questions, provide them feedback. This isn't in the script, but Ambrosia, there's no way I'm going to go on without thanking our incredible team at Komen Orange County under the leadership, I don't know why I'm getting choked up, but I could not be more proud of Ambrosia and everything she's done, the passion she has put into this. This is a huge day for all of us, and I want to thank Ambrosia for all she's done. Woo-hoo! And she would be really mad at me, and I'm not sure if we're live streaming, but Megan, sorry, I have to do this too. Our new CEO of Komen Orange County, Megan Klink. jumped in, worked with the team on this. She is recovering from a very major surgery. She's going to be fine, but in order to be back with us and be our best, she's at home recovering right now, and we'll see her back in a couple weeks. But when she is back, if you have not had the chance to meet her, please do, or reach out to us and say you want to come meet her. She's wonderful, and she's here with us in spirit as well. So also, I'd like to acknowledge the remarkable support that we received from our local Orange County hospital systems and nonprofit partners, from providing experts to do the breakouts, to helping us promote the conference, and also helping us financially put on this event. Today, 20 local experts from Orange County will be conducting sessions and providing you with the latest and greatest information and resources. If you're one of our presenters and you're able, please stand. Thank you. Thank you to our exhibitors in the expo also. I've got some homework to do. I want to go check all that out. Please take advantage of their resources and goodies. Okay, so finally, without the generous financial support and donations from the following businesses and nonprofits, there's one that's a nonprofit, I know it because I used to run it, this event would not be possible. So big, huge thanks to these groups that came together with us. Pfizer, Lilly Oncology, Celgene, Eastside, the Employees Community Fund of Boeing, the Chao Family Comprehensive Cancer Center, and Experian, who supported this conference and future programming throughout 2019. And speaking of Experian, we have a wonderful representative from Experian here today, Carol Urton. She is the Director of Corporate Responsibility and Sustainability for Experian's North American region that includes US, Canada, Chile, and Costa Rica. She's responsible for Experian's gift-in-kind projects, grants, employee donations, and matching programs, charitable activities, and she supports Experian's national consumer education programs. She is a published author, accomplished speaker whose goal is to inspire people to achieve their full potential during hardship in life. And Carol, would you like to come up?
Well, good morning. I had some prepared remarks, but I have to say, when I walked in through the door and saw this actually happening and all the wonderful people here, my heart was just overjoyed and overwhelmed. So it truly is an honor and a privilege to be here today. We're here today because we know that there's a direct link between physical health and financial health, and so Experian was very excited to be able to support this event. Every day in my job, I get to help Experian invest in our communities and help those out there in the world. So I have a direct link through my professional experience, but I also have a very personal connection to this cause. Over the years, I have watched my cousin, and I call her my cousin, but she's really my best friend. And I've watched her go through surgeries and treatments and just fighting this very fast-moving disease. I am super excited to say that my cousin Christy is here with us today. She's doing very well, and I would just like to, Christy, would you stand up and give a little wave? So it gives me great joy to see her here today, and you as well. Experian is the leading information technology company. During life's big moments, whether you're buying a car, sending a child to college, buying a new home, we're here to help you make the best out of your data and help you get to where you want to go. So in addition to supporting today's conference, we have some information that can help give you just some basic tools and understanding about credit and how to possibly improve it. So we're excited to be here to have a little short presentation around that. At Experian, we're committed to creating a better tomorrow, and we're really hoping today to help you create a better tomorrow as well. Thank you.
Thank you, Carol. And it is my honor to introduce someone many of you may know, and if you don't, I recommend you do. She has been assertive and persistent in her efforts to bring attention to the unique needs of the metastatic community in Orange County. With that said, she is the driving force behind this conference. Sandy Spivey has been involved in many local and national breast cancer organizations since her stage 2 diagnosis in 1995 at age 42. After being diagnosed with metastatic stage 4 breast cancer to her bones in 1998, she began her quest to learn more about the science of breast cancer so she could further influence research, and we are so thankful that she did. Sandy has participated side by side with scientists and oncologists on over 24 peer review panels for breast cancer research projects. She is a member of UCI's breast disease orientation team, where she represents patient voices. Please join me in welcoming and thanking Sandy, and from me too, your conference chair and MC, Sandy Spivey.
Thank you very much. I appreciate it. It's not often you get to get recognized for being alive. So thank you. I love to be alive. It's awesome. One thing that I want to mention this morning is I didn't have to worry about diarrhea or constipation this morning. And for those of you who are on any kind of treatment, you know how awesome that is. It's just awesome. I don't have to worry about those things. I do have to worry about my nose running, and I'm concerned about yours as well. Anyway, it's so exciting to see so many of you here at the conference. And when Ambrosia and I were talking about this conference, she kind of looked at me with big eyes and she said, how many people do you think would come? And people were saying, like, 75, 50, 100. And I said, no, 300 people will come. And she goes, what? And her eyes just like huge, like, wow, and here you are. We're here together. That's awesome. I'm so excited. We're not only here together in this room, but we're here virtually. There are many people who are watching this live streamed, and it will be recorded, so you can see this later on if by chance you have chemo brain and you forget some things. You can see this again. I mean, not that anybody would have that, correct? Or not that anybody, is of the age where they forget things, like age 25 and up. Anyway, we're so glad that you're here today, but let's see who is in the room today. So if you're from the medical community, will you please rise? Medical community people, if you identify medical community, please stand. Thank you so much for being here. We really appreciate your support. It's great. How many of those in the room are here to support someone living with metastatic breast cancer? Please rise. If you're here to support someone living with metastatic breast cancer. That is so awesome. We can't live a quality life without you, so thank you very much. Now please, if you've ever received any kind of breast cancer diagnosis, any kind, stage 0 through 4, please rise. And if you've received a diagnosis of metastatic breast cancer, please stay standing. Metastatic. Thank you so much for coming. I know it's very difficult. Remain standing or stand up if you have if you've been living with metastatic breast cancer for 3 years or more. 3 years or more. That's the ceiling we need to break, the 3-year mark. If you've lived with metastatic breast cancer 5 years or more, please stand. 5 years or more. Awesome. How about 10 years or more? You can see it is possible. It is possible. For those of us fairly new to living with metastatic breast cancer, be sure to connect with people in the room who have been living with it for a while And they may have worked through some situations that you're dealing with today. So please just go up and say hi. You'll see the blue or the teal ribbon that says living with metastatic breast cancer. Of course, some days we don't feel like we're living with it. We feel like we're just like managing the best we can through it, but you know, that's how it is. So please connect with each other, because that's why we're here today. I've been living with metastatic breast cancer since 1998, and that was 3 years after my stage 2 breast cancer diagnosis. My oncologist since 1998 has been Dr. Farah Brasfield from Kaiser Permanente in Anaheim, and she's going to be presenting the HER2 positive breakout for this meeting at 11 o'clock. And so she's awesome. You can see this is my chart of all the treatments I've been on. Under her care, I've gone through 23 types of treatment, some of them in combination, some of them repeated. The 8-foot document depicts the timelines of all the things I've been on since 1995, and you can see it in the art display behind you. It's up in the back if you want a closer look. I've spent most of my time advocating for metastatic breast cancer at the national level, and have participated on many national peer reviews and committees and various meetings and blah, blah, blah. But I felt it was time to come back to Orange County, my community, the place where I spent my childhood and most of my adult life. And it was time to connect with those who not only live with metastatic breast cancer, but those who support them either through treatment, emotional support, or even financial support. When you're diagnosed with early stage breast cancer, it's quite a shock. Then you found out how many people have walked that path. You finally find the medical and social resources to help you slog through it. Then you believe, or at least you hope you believe, it's over, save for taking medication that may keep it at bay. But when you're slapped in the face with a diagnosis of metastatic breast cancer, either after early stage treatment or initially receiving a diagnosis of breast cancer, because you can be originally diagnosed stage 4, managing through the first few days, weeks, and months can be challenging and overwhelming. You wonder what you did wrong. You're too young, you're too healthy, you're too busy to face the enormity of metastatic breast cancer. You have a family that needs you, and you have things you want to accomplish. You have a life that you've envisioned, but that life has changed. The resources for patients with metastatic breast cancer seem limited, as did the variety of emotional support. But we want to change that. In the greater Orange County area, we aim to build a community of those living with metastatic breast cancer so we can share and learn from one another. The Orange County Komen plans to put on other events throughout the year and other years in the future to bring the community together, and these will be designed to meet your needs, and that's why we need your evaluations. We want to find out from you the kind of things you'd like to learn more about. Some have said that Komen National and their local affiliates have had a singular focus on the victorious survivors with early stage disease and their motivational stories. Monies have followed along with that focus. But we know people are hyper aware of early stage breast cancer and mammograms. However, they're not aware of the type of breast cancer that kills, metastatic breast cancer. We as a metastatic breast cancer community with Komen support are demanding more research funding and emphasis on finding ways to extend both the quantity and quality of life for those living with metastatic breast cancer. Your attendance and support at events like this will help make changes. Komen is listening. One of our, I have the honor right now to talk about one of our local metastatic breast cancer research champions, Dr. Devon Lawson. And I met Dr. Lawson, and I call her Devon, and I'm sure she would rather be called that anyway, at a Komen event where, excuse me, I interrupted her presentation a number of times with a lot of questions. After, I do that, I don't know why. After the meeting, Devon suggested that I ought to be a presenter. I laughed, but she was serious. From that encounter, Devon has asked me to participate and give feedback on her research proposals and invited me to be part of the UC Irvine breast cancer disease orientation team so that I can share patient viewpoints on various topics that they discuss. Devon has provided her time and energy to solving biological mysteries of metastatic breast cancer. And boy, are there a lot of mysteries, a lot of things we don't know. I'm not going to read her formal bio. You can read it, and these are the people who went with us on a tour of her lab. Some of you are in the room, right? Who went on the tour? Who's in it? Yay, Catherine! Yay, Rebecca! Yay! But anyway, she and her lab team are working on ways of figuring out why breast cancer becomes metastatic, how to prevent that, what are the markers involved in that, and that perhaps someday we can have some kind of a treatment or a series of treatments that can help us live longer lives or even maybe not have metastatic breast cancer anymore. So she is one of our champions in Orange County, and I really appreciate her quite a bit. And right now I'd like to introduce Dr. Devon Lawson.
Good morning, everyone. My name's Devon Lawson, and I'm an assistant professor here at the University of California at Irvine in the Chao Family Comprehensive Cancer Center. And when we started planning this meeting, we thought that it would be a good idea to start off the morning with some basic, trying to introduce some basic concepts about breast cancer and metastatic breast cancer so that we can kind of all get up to speed and on the same page so that we can get as much as possible out of the more detailed sessions that are going to follow later on in the day. So I'm going to start basically by starting really, really simple and kind of tell us a little bit about breast cancer, how it starts, how it becomes metastatic, why it's important to you clinically and in terms of the way that we treat metastatic breast cancer. And then as things go along, I'll tell you about some new discoveries and new promising therapies, things that I think are going to really make a big difference in the next year or two in terms of being able to treat metastatic breast cancer. Okay, so for those of you who've never been here before, you guys are actually sitting on our main education campus here at UC Irvine. And UC Irvine, our cancer center is a little bit special. There are a couple of things that make it unique compared to local hospitals and community centers. So UC Irvine is actually an academic medical center. So that means that we have these three really important missions, which you can see up here: discover, teach, and heal. And basically what we do is we have dedicated scientists in buildings like this one here, where we do detailed experimental analyses in order to bring about new discoveries that are important to potentially treat metastatic breast cancer in new ways. We also teach tomorrow's scientists and doctors to hopefully propel forward better care for patients in the future. And then lastly, of course, as you know, our main goal is to bring new therapies and actually treat metastatic breast cancer patients in general. And as an academic medical center, we do this by teaming up scientists with doctors and clinicians, essentially, to provide the most innovative new therapies coming out every day. And so for those of you who are interested potentially in clinical trials, University of California, Irvine has a number of clinical trials with new therapies that are just coming out to treat metastatic patients. So please come to our website, or I can tell you more about it later, and I can tell you more about some of those new therapies that we're working on. Another important point about UC Irvine is that we're actually a comprehensive cancer center. So that means that of all cancer centers in the United States, there are only 49 cancer centers that have been designated by the National Cancer Institute as meeting this really important level of excellence as well as breadth for cancer therapy. So just another important point about our cancer center and being able to hopefully provide you with the best therapies in the future. Okay, but with that, what I'd like to do is start again, like I said, by bringing you guys a little bit more information about basic biological concepts of breast cancer. I'm going to tell you a little bit about what causes it in very basic terms. I'm going to tell you why there are different breast cancer subtypes, and this is going to be really important because you can see on your agendas that later we're going to break off into different sections depending on which subtype you either have, your family member has, or you're most interested in learning more about. So I'm going to tell you about how these subtypes come about. I'm going to tell you a little bit more about metastasis and how it initiates. And then lastly, I'm going to tell you some new discoveries coming out in this field. Okay, so what causes breast cancer? So in order to understand what causes breast cancer, we have to know a little bit about the breast. So this is a schematic representation here, basically, of human breast tissue. Some of you have probably seen this before. But basically, the breast is a ductal organ, which you can see here, comprised of these lobular structures. Some of these terms will probably start to sound familiar for those of you who are used to hearing them in terms of your clinical diagnoses and histological, pathological data from your clinicians. But the breast basically is comprised of these lobular structures here, which essentially are balls of cells that function to secrete milk during lactation to feed our young. Those milk proteins essentially are distributed down into these ducts here, which connect to the nipple, that enable feeding of the offspring. And then these ductal structures are essentially surrounded by these adipocytes or fat cells, which instruct those epithelial cells or those milk-producing cells how to function. So taking a closer look here, basically, let me see if I can move this so I can actually see my slides. Here we go. So basically, if you take a closer up image, essentially, of one of these guys here, one of these lobular structures, this is sort of what it looks like in 3 dimensions. And what they've done in this image is they've kind of sliced off the top so you can see inside this ball-like structure. And so what's important here is you see that this structure is comprised of this basically epithelial cell layer here, which again, they secrete the milk proteins during lactation. And those cells are surrounded by these orange cells, which you can see here that look kind of like these little spider-like cells. And these cells are called myoepithelial or basal cells, and they actually have this contractile function, a lot like for example, heart or muscle cells, which basically push the milk through the ducts and down towards the nipple. And what's important about this is that these ducts and lobules are made up of millions and millions of cells. And this is important because the breast is essentially an extremely dynamic organ, so it's constantly remodeling itself during different stages of our life cycle. For example, menstrual cycles, during pregnancy, and also during menopause. The gland is constantly growing and shrinking. So for example, if you take this example of the breast, again, the same sort of anatomical diagram that I just showed you a minute ago, you can see before pregnancy, basically it looks like this, where you have, again, these little lobular structures here that release milk into these ducts. When we become pregnant, what happens is these lobular structures start to grow. So the cells really will divide and start to grow. So you have millions of more cells that are then able, when we start lactating to feed our young, we are then able to basically produce more milk to feed the young, right? So you can see how these lobular structures really grow dramatically. I'm telling you all this because in terms of breast cancer, all of this cell division, this constant growing and shrinking, really makes the gland very susceptible to developing cancer. I'm going to tell you a little bit more about that. So every time a cell divides, it basically opens itself up for being able to sustain DNA damage and mutations that can cause cancer. So if you take this example here of this purple cell, where you have its genome represented here, so the DNA in this black structure, when this cell starts to divide, it copies its entire genome. So that's all the DNA and base pairs in that cell. And occasionally what happens is when it copies the genome, it makes a mistake. And you get introduction of the wrong base pair at this potential site here shown in this schematic. What happens is when this cell actually divides, it gives one of the genome copies to this cell and one of the genome copies to the other cell. This cell now essentially is a mutant cell in that it has this one mutation error here. And what happens when these cells continue to divide, right? Because cells continue to divide and shrink throughout our lives. It passes on this mutation to all of the daughter cells that it makes throughout the rest of your lifespan. And again, why I'm telling you this is to try and illustrate this concept of how we can start to develop cancer. So I'm going to rattle off some numbers here just to give you an idea of how often this happens in our bodies every day. So the human body is made up of 37 trillion cells. So for those of you who like numbers, that's a 10 with 12 zeros after it. Every cell has 3 billion base pairs of DNA, so that's, again, a 1 with 8 zeros after it. That's more money than, like, you know, Donald Trump and Zuckerberg combined. So the error rate of repair, or the error rate when our cells divide, essentially, or copy their genome, is 1 base in every 10 billion base pairs that are replicated. So again, about 1 in 10 to the 9. And so since our bodies make 50 billion cells on average every day, you can imagine that if you're mutating at this rate, about 1 in every 4 new cells has a mutation in it. So why don't we all get cancer all the time? Well, we kind of do, I guess is an interesting thing. But our cells have really well-established, very rigorous repair machinery within every cell that enables them to essentially repair these little mutations and errors. And we also have a very important immune system, right, which helps us recognize cells that have these mistakes in them and then kill them off. But as you can imagine, if we're having errors like this often during replication, these mutations will occasionally escape our repair mechanisms and be passed on to cells for future generations. So this is basically what causes our cancer essentially to initiate. So remember I showed you those ductal structures and those lobular structures in the first slide. So this is what one of those ducts look like. And again, they're supposed to be comprised of this single layer of cells, right? Here, breast epithelial cells. But what happens is if one of these cells sustains one of these mutations in a gene that controls cell growth, so our genes control how our cells behave, what can happen is the regulatory mechanisms that keep this single layer here from staying a single layer can start to go wrong, and the cells can start to divide essentially uncontrollably and produce things like what we call DCIS, so ductal carcinoma in situ, where you have all of these cells here piling up in the center of the duct where they don't belong. And then for those of us sitting in the room with metastatic disease, occasionally what happens is as these cells start to divide uncontrollably, they breach through these ducts and start to invade into the tissue and then spread to other parts of the body. So the take-home message that I wanted you to get out of this is that breast cancer is essentially a genetic disease caused by DNA mutations that are generated during this cell division process. So no matter what anybody tells you out there about diet and exercise and eating too many cookies or too much red meat, I can guarantee you that your cancer is not your fault. It was not caused by something that you did wrong in terms of your lifestyle. It is inordinately caused by this process of cell replication, which we all have to do in order to live, okay? It's not to say that your diet and lifestyle can't help improve your immune system and make sure that your DNA repair pathways are acting properly, but it's really important to understand, because I seem to get this question a lot about what did I do wrong? This is how it happens, and it's not because you did something wrong. Okay, so let's move on to what are the different breast cancer subtypes. So this is really important for understanding why we have different types of breast cancer and why they're treated so differently. So back to this little diagram that I started with in the beginning with the breast here. And you can see essentially that we have these lobular structures that empty into the ductal structures here. So if you take one of these lobular structures here that's circled here, and you actually just take the ball here and you cut straight through it, what happens is you get sort of a circular structure that looks something like this, where you can see, again, this single layer of cells here called luminal cells, which secrete the milk into the center of the duct, during lactation. So these luminal cells here, basically they secrete milk, and they have 2 really important receptors expressed on their cell surface here in the center. One is called the estrogen receptor, and the other one is called the progesterone receptor. These sound familiar to anybody? Of course, right? Another interesting cell type, so around this single layer of luminal cells are these basal cells here, or myoepithelial cells in blue. And these cells are different. Again, these are those contractile cells that squeeze the milk down through the ducts. These basal cells predominantly are negative for these 2 receptors, so they do not have these receptors expressed inside of them. So you can probably start to imagine that these different cell types, since they have these different properties, are basically the cause of why sometimes we get basal-like cancer, which doesn't have these receptors, and sometimes we get the luminal-like cancers, which do have the receptors. And I'll tell you a little bit more about that in a minute. HER2, for those of you who have HER2 cancer, is a little bit more complicated because the HER2 receptor can be expressed in both cell types, and we sometimes see it in basal-like breast cancers and luminal-like breast cancers. And I'll mention that more in a minute too. So basically, going back to our thing about mutations and how they cause breast cancer, when you get mutations in these cells, essentially you get a more luminal-like breast cancer. And when you get mutations in these cells, or cells sort of like them, it's a little bit more complicated than this, you'll get a more basal-like breast cancer that doesn't express these two receptors. And this diagram, I don't want to go into too much detail, but for those of you in the room who have thought about this a lot are really interested in this idea, so some of our more advanced folks in the room, it is a little bit more complicated than this. So what's important to note from this slide is, again, you see these cells here, these luminal cells, as well as our myoepithelial and basal cells. And scientists use these charts to understand how cells develop in the gland and basically where these cells come from. And we think that they come from these stem cells over here, which basically will proliferate, like we talked about it a few minutes ago, and they'll divide and divide and divide, and eventually they differentiate into these different cell types. And what we think causes these different subtypes of breast cancer, again, basal, HER2, and the luminal subtypes, and for those of you who may have a Claudin-Low subtype, basically what we think happens is that when you get mutations in specific types of genes, we talked about growth regulating genes, but there are all kinds of genes which can be involved in cancer initiation. In different cells at these different stages along this differentiation process, the output is basically cancers that look very different and express, different hormone receptors. So whether you understand that or not, it's not important. That's just to, if you're interested in the biology of how this happens and understanding, why it is that we get these different subtypes, it's okay. What's most important to understand is that there are 4 major subtypes. There are these luminal subtypes here, this one here, and this one here. They express the estrogen receptor and the progesterone receptor, so the hormone receptors, HR. Some of them are HER2 negative and some of them are HER2 positive. And then we also have the triple negative breast cancers, which are basically basal-like breast cancers, and they don't have these hormone receptors. Sometimes they also have the HER2 down here, and sometimes they don't. And you can see that most breast cancers essentially fall into this category here of being luminal-like and having these estrogen and progesterone receptors. So why am I telling you all this? Why are these receptors so important? So some of you probably already know this. But basically, the estrogen and the progesterone receptors are really important for how we treat this type of cancer. So these molecules, or these proteins, basically they bind to estrogens, which float around in our blood. And we talked a lot about pregnancy, right, and how the gland grows and shrinks. And that usually happens because our cells, our receptors, recognize these hormones in the blood as changing during pregnancy, right? So then they start to grow. So what we do essentially, so basically this is what the estrogen and progesterone receptor do, essentially. When we bind to estrogen receptor with the molecule, we start to get growth of the cell. HER2 functions in a fairly similar manner, where essentially you have a HER2 receptor on the cell surface. When it binds to its growth factor, basically outside of the cell, you get this whole series of reactions that happen, and then again the cell starts to grow. So basically both of these pathways, the hormone receptors and the HER2, cause cells to grow when they're activated. And so in cancer, why do we give you tamoxifen or other hormone blockers? Essentially because what happens is tamoxifen pretends like it's estrogen, and it binds to the estrogen receptors, but rather than binding to them and causing the cell to proliferate, it actually blocks the ability of estrogens to bind, and then we stop the cells from growing. So that's the mechanism of action of these of drugs that block the estrogen receptor. HER2 is similar in some ways. So basically the main drug for the HER2 cancers is called Herceptin. And again, we have these proteins on the surface of our cells. This is a cell membrane, basically depicted here. And the HER2 receptor is shown in purple. When it binds to its growth factor, the cells start to grow. And Herceptin is essentially an antibody which binds to these receptors here and prevents them from being able to transmit this signal. This is also a little bit controversial in recent years, and I'd be happy to tell you more about that as well. But this is what we think is the main way in which Herceptin works to block growth from the HER2 receptor. Okay, and then for those of us who have triple negative breast cancer, you're probably wondering, okay, well, what am I supposed to do? And the unfortunate fact is that we don't have any really great receptors for the triple negative breast cancers. We don't have, these cells don't have estrogen, they don't have progesterone receptor, and they don't have HER2. So what we're left with is being able to target them with a much less targeted approach. And we use these different types of chemotherapies, essentially. So paclitaxel is a first-line chemotherapy for triple-negative breast cancer. And the chemotherapies work really differently. Rather than trying to hit a receptor on the surface of the cell, they basically screw up the cell's biology so that the cells can't divide anymore. And then the cells start to die because they can't divide. And so, as many of you probably know, this can be very toxic to a lot of other cells in our body who are wanting to divide, like our skin and our hair and other things. And so, in moving forward for this subtype of breast cancer, we're really focusing on trying to find better targeted therapies so we don't have to come in with such a abrasive and non-specific approach for targeting them. Okay, so let's move on to what is metastasis. How are we doing on time? I didn't see a time thing. What do we have left here? 15 minutes, thank you. Okay, so I'm getting close to finishing up. Is that 15 minutes for questions too, or? Okay, so just to move into what is metastasis. So I always like to start this with telling you a little bit about how metastasis happens, because I feel like sometimes there's a little bit of a misconception. So when you get metastatic, when you're diagnosed with a metastatic tumor in the bone or the brain or the lungs or the liver, it's not because a new cancer grew from the cells in the lung. It's actually quite the opposite. What happens is the tumor in the breast essentially uses the body's highway system, so these blood vessels as well as these lymphatic vessels that carry immune cells and also cells that oxygenate our blood. Basically, these cells, as I mentioned, they breach out through the ducts of the breast and they start to go into these blood vessels and actually travel through the blood vessels to other parts of the body. So what's important to understand about metastasis is these are cells that are motile. They actually are traveling. And here's just another depiction of, as scientists, what we study when we try and understand metastasis. A metastatic cell has to go through all these different processes in order to get into the blood vessels. They have to crawl out of the blood vessels. They have to go to the liver and figure out how to survive in a new organ that really they're not used to and doesn't make any sense. And so for research, what we do is we focus on understanding how each of these steps happens so that we can figure out how to block these things from happening. So my lab in particular, and I'm going to finish up in the last couple of minutes by telling you about a couple of new discoveries in my lab that also are relevant for what other people have found. So research in my laboratory here at UC Irvine focuses on trying to identify which kinds of breast cancer cells are going to metastasize. So we talked about DNA mutations and how they cause breast cancer, right? And how all the daughter cells of a cell that's mutated will all have this mutation. Well, what happens over time is that the cell accumulates more and more and more mutations. So our primary tumor in our breast actually is comprised of all these cells that have different properties. They have different mutations, they have different functions, they behave really differently. And what we really want to understand is which of these cells are the ones that are actually going to make metastasis, because we want to understand what's unique about those cells and their basic function in biology so we can stop them from making metastatic tumors, and also so we can kill them when they are in metastatic organs. So we use this approach called patient-derived xenograft models, where we basically take patient samples and we make little mouse avatars out of them. So we put your tumor essentially into a mouse mammary gland or breast, and then when the tumor grows, we'll transplant it into subsequent generations of mice so that we can study how the cancer cells from your own tumor essentially metastasize to other organs in the mouse. So this is just a picture of a metastatic colony, essentially, in the lung of one of these animals. So for those of you who have seen your own histological or pathological slides, this is basically the lung tissue here. So you can see a mouse lung and a mouse lung schematic. This is the lung tissue. And then the cancer cells are basically right here in the center. They look a little bit differently. They tend to be a little bit bigger. So to understand the function of these, what makes these metastatic cells different, essentially, than all those other cancer cells that don't metastasize, we basically use this very special type of procedure. So for those of you who remember from high school biology or whatever, there's this thing called the central dogma in biology, right? Where essentially DNA is the blueprints. They make this molecule called RNA, which is essentially the manual, the instruction manual for how to build the cell. And those are translated into proteins, which actually are the building blocks that make the cell or the house, if you want to think of it as like architectural blueprints, the instruction manual, and building blocks. And so what we do is we take these single metastatic cells from the lung, we extract all the RNA, which is basically instructions for what makes the cell do what it does. We amplify up the RNA so that we can sequence it and understand what the instruction manual is telling us. And we use this really high throughput sequencing technology to basically get us understanding of how these cells are functioning. Okay, so an interesting thing about genome sequencing and sequencing in general is that I like to tell people about how far we've come in terms of advancing this technology. So in 2003, as a scientific community, we completed what's called the Human Genome Project. It took 13 years to sequence one genome, so it was started in 1990. Hundreds of labs worldwide worked on this amazing project. We sequenced 3.3 billion base pairs, the number probably sounds familiar from earlier, and it cost $3 billion to finish this project. So what's really cool is in 2019, we can sequence next to the entire genome, most of the genome of a single cell, for like $100 or a few $100 in one lab. So we've really come a long way in terms of being able to understand what's going on in individual cells when they're metastasizing. And we use these nifty little procedures basically to capture our single cells. So here is a droplet-based technology, and here's a microfluidics-based technology where we actually capture little cells in these fluidic chambers, and then we prepare the cells essentially for sequencing. And this is how the sequencing technology works. We have these really cool glass slides, or flow cells, where all the RNA molecules are captured on these oligo chips. Without going into too much detail, we can then read out the entire sequence from single cells with these fluorescent molecules, which basically represent the addition of new base pairs as we're sequencing. So in my lab, what we found when we looked at 3 different patient models, we compared tumor cells and metastatic cells for what's different about their gene expression. We found 330 genes that were different between the metastatic cells and the primary tumor cells. And one really important thing when we looked at all those 330 genes that came out of it was that we found that our metastatic cells have this really special property compared to the primary tumor cells, where they can actually inhibit immune cells. And when we looked closer, we realized it was a very special type of immune cell called natural killer cells. Who's heard of natural killer cells? Yeah. So these cells are really, really important in our bodies because they basically roam around the body and they find infected cells or damaged cells like cancer cells and they kill them. And so what we found is that our metastatic cells have grown really smart. Essentially, they've upregulated proteins on their surface that enable them to figure out how to block these immune cells from killing them. And so we've teamed up with a group at UCSD, Dan Kaufman's lab, essentially. And what we're trying to do is engineer super NK cells. So we actually will take new NK cells and transplant them into patients, or in this case, into mice, to try and clear out those metastatic cells that have generated this new way of suppressing them. So you can see in this example with these cohorts of mice, when they were transplanted with cervical cancers, so you see the signal here of the cancer growing over time. When we transplanted in these super NK cells, you can see that we were able to actually kill off most of the metastatic or disseminated cervical cancer. So this is really nice work done by Dan Kaufman's group at UCSD. And now we're trying to see whether we can use a similar approach in humans to target and destroy metastatic breast cancer cells. And this type of approach, these NK cells, have been hailed by Science and other journals in the last few months as being basically the next great cancer immunotherapy, potentially. Because unlike T cells, we don't really have to get them out of you, specifically your T cells, and put them back in. We can just grow them out of a dish and put them into patients with much less cost and difficulty in doing so. And I think I'm going to go ahead and wrap up here, but I'm really excited about this new avenue of cancer therapy, these natural killer cells. And here I just wanted to list that there are tons of new clinical trials coming out all the time using different strategies to reawaken these natural killer cells so they can fight different kinds of disease. So here, the one I mentioned was this adoptive cell therapy approach, where we're actually transplanting new NK cells that aren't inhibited. But there are a lot of other trials going on where they just are trying to use small molecules and drugs to basically improve your own NK cells and their function. So here are some trials, and this paper has a lot of different trials, so here's some other different ways to uninhibit NK cells using checkpoint inhibitors or target immune suppressive pathways. And so I think with that, I'll go ahead and finish so I can answer some questions. But I did want to thank everyone, of course, in my lab and other labs who've been working on this. So these are some folks right here down the street at UC Irvine that did a lot of work on some of these projects in my lab. One of them just got married last year, Ryan, who was really important in the NK cell study. And with that, this is actually our anti-cancer challenge that we do every year. I believe Brian in the back, who's got his hand up, has a booth telling you about this wonderful new project that we run every year to raise also money for cancer in general. And you see we're a breast cancer lab, so we have some funny looking pink items on for this challenge. But with that, if you guys have any questions, I'd be really happy to take a few questions now, and I'm also will be here the rest of the day to take questions. Well, there's a lot of reasons that it could be, and I think what's most important to understand is that as scientists, we don't know always. And that's one of the main things we're trying to figure out, is why this happens. I think probably the most likely reason is we failed to actually kill all the cells in the metastatic tumor. So remember that picture I showed with all those different kinds of cancer cells in different colors? So a lot of times these therapies will only target like 75% of them and leave the other ones behind, because the other ones are a little bit different and the drug doesn't hit them. And so what happens is most of the cancer dies, and then those few cells, like maybe there's 100 cells left, cancer cells, over time they just grow back. And then we have to find a new way to kill them because the first drug was not effective against them. So I think it's probably, in most cases, it's this. In other cases, it can be that these metastatic tumors will actually send cells out themselves through the blood, right, and make new tumors. And so we may kill one tumor, and then 2 years later, we realize that there was another tumor growing that we didn't see, and that one is now a new tumor we have to deal with. So, great question. So if you're interested in the natural killer cell therapies, as well as there's all kinds of these adoptive cell therapies, there are ways to look online for trials. So they're already available, but you have to get your clinician or your oncologist to enroll you in a trial that you're eligible for. So the short answer is you can get the therapy now if you can find a location that you can go to and you meet the trial criteria. Beyond that, some of them are looking really promising. I would expect them to start becoming more available if they're successful in the next few years, like, you know, without having to be on a clinical trial. So the cancer cells, they have all these proteins that allow them to communicate with other cells in the body, right? And the one that I mentioned was cancers often, in this case that when we were looking, it looked like the metastatic cells learned how to suppress immune cells, right, which allows them to survive in metastatic organs. But cancer cells can do other things. You know, they're bad actors, right? They're secreting all kinds of things that they're not supposed to be doing because of all this genetic mutation. And a lot of times that causes immune cells to go to those cells because they recognize that there's something wrong there and start to try and kill them off. And so if you have an inflammatory cancer, probably your immune system is trying to kill off the cancer. And sometimes it's successful, and other times it just causes tons of immune cells to go there and try to kill it. But, you know, they start to make damage too because you have all this immune reaction happening to the tumor. So that's a good question, but it's definitely part of the same issue. Great, well, thank you so much.
Thank you, Devon. So now we've learned that breast cancer is more complex than we thought. It's really, really social. It likes to get to know the normal cells around it and hide. and you know, and try to grow and use the supplies that are around it to grow, and taking them away from normal cells. And you know, they're evil, but they're really, really friendly, and they really, really, you know, like your other cells because it helps them grow and they make them die. And you know, so you got some really friendly kind of, you know, whatever, frenemies, I guess, inside of you. So now we know that, you know, a little bit about metastatic breast cancer and just breast cancer in general. Every time Devon speaks, I always learn something new, and I took some notes today because there's some things that it was like, oh, that's how that works. Okay, so I'm really glad that we had that. So let's learn more about metastatic breast cancer research. And what I'd like to do now is introduce Krissa Smith. She's the Director of Research Programs at Susan G. Komen headquarters. She lives in San Diego, but the headquarters is in Dallas, so she can explain that. I won't explain it. She directs and oversees the implementation of Komen's research programs development. So the money that you put toward research goes through her department, that goes to the you know, through various channels in order to get to the right person to do the research. She's responsible for developing strategies and priorities, as well as managing a $25 million budget to the grants that are awarded every year. So please join me in welcoming Krissa Smith. Thank you.
Well, thank you so much for having me today. I'm so pleased to see so many of you here. Can everyone hear me? If anyone needs a seat, there's a beautiful table right here in the front, if you'd like to join us up here. I'm so honored to be here with you today, to be a part of Komen at this really transformational time. I feel incredibly connected to this community and to this work. Before I came to Komen, I was a researcher in the lab. Like Devon, can you guys hear me? Am I being loud enough? Okay. Like Devon, and I worked on those breast cancer cells that are crawling out of the blood vessels and looking at those immune cells and supporting cells around the blood vessel to try and stop them from crawling out. Devon showed some really great pictures. Thank you for that, for setting us up with that basic understanding of breast cancer today. My path to this research started many, many years ago. When I was 5, I was still really cute. I went to go visit my grandmother in the hospital, and that would be the last time that I would get to see her. What I didn't know then, that I now know, is that she was dying from metastasis to her brain from her breast cancer. And my dad, who was a young resident, a doctor in training at the time, had snuck me in so that she could say goodbye. The room was really dark. There were tubes coming out of her. I was snuggling on the bed with her. Really didn't understand what was happening. But it really made a deep impact on me. My father was training to be a radiologist because he was driven to medicine because my family needed an expert and we needed an advocate. My grandmother, when she was living in rural Idaho, at the age of 38, in the early 60s, had a radical mastectomy. She had 8 young children. It was a week before Christmas, and my father was 8 years old. So this is a part of my family. This is a part of my story, and I stand here with you today. My father has now lost 4 of his 7 sisters to metastasis. Not all of it was breast cancer. Our family has that DNA repair process that Devon was talking about that's so essential. It is very essential. We have a bad gene, one of those genes in our family that has a mutation. And so 4 of my aunts, we've lost to metastasis. I really hate cancer. Sorry. So this really deeply impacted me. So I wanted to fight breast cancer. I wanted to know more and be able to be a part of this. I started out in the lab, and now I feel very fortunate to have the opportunity to have an impact at a national level working with Komen and their research programs, and work with some really amazing people, some of whom are speaking today. So I'm going to echo some of Sandy's comments right now. In the past few years, we've witnessed a significant change in the breast cancer community. through the emergence of strong voices of all of you, the metastatic breast cancer advocates. I'm personally so grateful for the guidance and leadership of some of these incredibly impactful and passionate advocates, such as Sandy Feinstone, who serves on our scientific advisory board, Kelly Shanahan, who's participating in the conference today, and the incomparable Sandy Spivey, who we've already heard from several times today, and if you stick around until the end, you'll get to hear from once more. These advocates have really worked alongside so many others to inspire our laser focus on the breast cancer that kills. We hear that word a lot, advocate. What do we usually think of? We usually think of Capitol Hill and going up to lobby Congress, right? But being an advocate is doing what all of you are doing. Showing up, standing up, and speaking up. Or more importantly, speaking for those in the community who can't, all for a cause that you deeply believe in. In the early days of breast cancer advocacy, this is exactly what happened. Advocates brought attention to the disease, they spoke out for the need for high-quality medical care, for better access, and they spoke about the need for increased federal research funding. Millions of women and men have been educated and empowered. But now, metastatic breast cancer advocates are driving this change in the breast cancer world. So much focus has been on the primary disease, ignoring the fact that the majority of women actually die from metastasis. Most of those experience metastases after doing everything that they thought was right, only to face recurrence or a metastatic disease. Komen stands alongside of you to change this with our focus on reducing mortality and with our focus on breast cancer metastasis. However, our ability to discover and deliver the cures for metastatic breast cancer relies on this continued investment in research. And I don't just say that because I work for the research programs department. This is really a vital part of finding the cures for breast cancer. We know so much more than we ever did when my grandmother had her radical mastectomy in 1964. But do we know enough? Clearly not. Dr. Danny Welch, one of our Komen scholars, he has been referred to as the godfather of breast cancer metastasis research. He talks about how we need this understanding of basic concepts and targets so that we have enough water pressure, so all those ideas pushing into a fire hose so that we can put out the fire for the next metastatic treatment. We always need to be ready for the next fire during that treatment course. And that comes with knowing more, finding better targets, and getting new treatments to patients in the clinic. We heard from Dr. Lawson today. She gave us a great overview. She also used a lot of acronyms. There are a lot of acronyms in cancer biology. In fact, it's not surprising to me that the DoD funds cancer research because I think they were perfectly at home with all the acronyms, right? It's a perfect fit. But what this means is the road to new and improved treatment starts with an understanding of that basic biology. We need to understand what is helping that breast cancer grow and metastasize. We need to understand how we can harness the power of our immune system. Devon is doing great work with that. We can develop treatments to cut off that growth, especially when it's growing in distant organs. The scientific community is unraveling that complex story, those mysteries, to help us deliver new therapies and achieve a significant reduction in mortality. And we've had some recent wins. Has anyone heard about the Impassion trial? The first immunotherapy was approved for breast cancer patients just a few weeks ago, which is incredibly exciting news. We've heard a lot about immunotherapy, and we've been hopeful, just like the questions we've heard today, that that was going to get to patients sooner rather than later, and it's finally getting there. And that was approved specifically for patients with advanced and metastatic disease. We've had other progressions moving forward. In a new Journal of Clinical Oncology publication, Komen-funded researchers found that a drug called neratinib, which inhibits many proteins including HER1 and HER2, it had increased activity against brain mets, another real challenge in the community, when it was given in combination with chemotherapy. The results of that study showed that there was significant reduction in brain mets size in roughly half of the patients treated. So we're making progress step by step. CDK4/6 inhibitors, has anybody heard of those? Everyone was excited about these, right? This was going to be the next thing that was going to fix everything. Turns out they were much more complicated. Science is very complicated. You saw a little bit of that earlier. We're seeing progress there so we can understand that those actually are effective treatments, but only for people in the right setting and only very personalized precision treatments. So you'll hear more about some of these wins from researchers presenting later today. But the number of men and women who are dying of breast cancer hasn't changed, and thus our work is not done. We need new treatments, but surprisingly, that journey doesn't often begin at a pharmaceutical company. It actually begins through funding from the National Cancer Institute organization and organizations like Komen that fuels the scientists that are priming that fire hose, bringing all of those ideas to funnel to get down to those treatments that will actually work. This is one of the reasons why we have strong advocacy for federal research funding, and it's so very critical. Our pharma colleagues need those biological breakthroughs to help find new treatments. In 2017, the government leaders approved $5.6 billion for funding at NCI. That's nearly a $368 million increase from the previous year. And that included about $545 million specifically for breast cancer research. Seems like great news. And it's an extraordinary amount of money. But with the increased expense of research and the impacts of inflation, this doesn't actually represent a growth in research investment. It actually shows a reduction in investment power. The total cost of getting a drug to market, some estimates are at $1.8 billion, and that's in 2009 dollars. It's a fraction of what we need to really get things to patients. There was a question about how long it takes to get to patients. It takes an average of about 12 years for a drug to travel from the research bench to our pharma partners, through clinical trials, to that finally gets to patients. So let's say there are 5,000 new ideas out there. We hear a lot of these. How many will make it to human testing, to phase 1 clinical trials? 100? Raise your hands if you think it's 100. Oh, you guys know too much. 50. 50? Okay, how about 10? 10, that's getting closer. The estimate is actually 5. 5 out of 5,000. And of those 5, how many are going to be approved for patients? 1, about 1. So if we look at the amount of ideas and things we need to invest in to start that process, to get to that one treatment that will save lives, it's an incredible amount of work. And we desperately need the dollars and the focus to do that. If we want to accelerate progress against metastatic breast cancer, now is the time. We need to continue to advocate for that increased research funding. But advocacy doesn't just happen on the Hill. Organizations like Komen, like Mediviver, like BCRF are also investing in research. And because of that advocacy, our organizations are specifically increasing investments in metastatic breast cancer research, coming away from that focus on the primary disease. This year, Komen's total investment in metastatic breast cancer, the total will become over $200 million. That's to more than 500 metastatic breast cancer grants and over 50 clinical trials. Because of our specific focus on metastatic breast cancer research in 2018, we funded over $12.8 million in metastatic breast cancer research grant, which represents over 50% of the grants funded for 2018. And that focus will continue for the coming year to increase our investment in metastatic breast cancer research to go upwards of $200 million. However, all of this funding will only be impactful if we have the best scientists working on these questions. We have a significant challenge in our scientific workforce that is specifically impacting our young scientists. I'm going to refer back to Devon again. I feel like we should be up here together. She's an example. Only about 20% of those who graduate with their PhD, not who start their PhD program, but actually graduate with a PhD, will actually achieve an academic research position that will allow them to compete for that federal funding, that $545 million. Those who do achieve the holy grail of a research position are faced with intense competition for research dollars. The success rate for receiving a grant from the NCI right now, do I take a vote again? More than 15%. Well, you're wrong, because it's 11.9%. Simply put, to be a young scientist aspiring to find the cures for metastatic breast cancer, it's a really tough journey. Komen believes that inspiring and investing in our brightest young minds is critical to progress against metastatic breast cancer. One of the goals of our research programs is to attract more of the best and the brightest researchers towards solving these really complex problems of metastasis to fuel the pipeline. Dr. Yibin Kang, one of our Komen scholars, I asked him what brought him to breast cancer metastasis, like why he had chosen to study that field. And he said that it was the absolute hardest research question that he could find, and that was why he wanted to tackle it. And he is one of the brightest people I've met, so I'm really grateful he's working on it. Our research program, where we've issued a call for research applications from the scientific community, is focused entirely on our young scientists and entirely on metastatic breast cancer. And we've followed that for the past 2 years. Now, we've talked about funding, and we've talked about the scientists, but we need to come back to you. We need you to advance our research efforts. And you might think I'm talking about clinical trials, and yes, that is one very critical, important aspect. We need to make them a normal part of our vocabulary. We need people to understand and feel comfortable about clinical trials before they're in crisis and they need them. So I'm going to make a challenge that everyone here, all 300 of you, or so, can talk to one person this week about clinical trials. You don't have to recruit them, but I'm sure if you did, there would be some really happy scientists and doctors trying to recruit to those clinical trials. But if we need to make this a part of our normal everyday speech, something like a physical or a vaccine, that clinical trials are part of our care from the very beginning. If you do have questions about clinical trials, if you aren't sure if it's for you, there is help. There are resources. Komen has a clinical trials helpline with oncology social workers, people who can help talk you through the process, answer any questions, get you to the right places. And breastcancertrials.org actually offers a matching service. So if you are looking for a clinical trial in your area, you can input different fields of information, and it can match you to something that might fit for you. It's an important, George Sledge, our chief scientific advisor, stated, "Research cures cancer, but only if the research can get from the laboratory to the clinic. And the only way this can happen is through clinical trials." Has anyone here participated in a clinical trial? Can we all give these people a round of applause? It is hard work. Thank you. Thank you for your participation in helping us move this work forward. It would not happen without the people, the heroes who participate in clinical trials. But what is also important to Komen is research advocates, including those who are living with metastatic breast cancer as partners in all aspects of our research program. This is what we believe, that the ultimate beneficiaries of our research investment, the patients, must be working alongside our scientific community every step of the way, from the strategy to the science in the trenches. So every research grant that Komen receives is reviewed by a Komen advocate in science. I'd like to specifically thank Sandy Spivey, Sandy Feinstone, and Andrea Hutton for participating in our peer review process, especially now with our focus on metastatic breast cancer. And most importantly, every grant application that we fund, every single grant, must have a patient advocate as a key member of the research team. For some of our scientists, especially our younger scientists working away in the lab, this may be the first person with metastatic breast cancer that they have ever met. Imagine the power. Imagine the passion driven by these personal connections and what progress can be made. We are so very grateful to our doctors, our scientists, our patients, and our advocates who have helped deliver more effective treatments. We've come a long way, but there's still a long way to go. Elevating the voice of the metastatic breast cancer community does not diminish other voices. Saying that those living with metastatic breast cancer, that they matter, does not diminish those who are walking a different journey. It means we want all women, to survive breast cancer. Komen is committed to this focus on metastatic breast cancer and will do whatever it takes to end this disease. Your voices have been heard, and we will continue to listen, to learn, and to better serve you. I am proud to stand beside you. Today, it is my absolute privilege to introduce one of the most brilliant breast cancer researchers of our time, Dr. Joe Gray from Oregon Health Sciences University. He has dedicated his life to answering the difficult questions about breast cancer and specifically looking for ways to make breast cancer and metastases more treatable. There is no better person to work on this problem. I mentioned that I used to work in the lab. What I didn't mention is that Joe Gray is kind of a superstar hero, especially for young scientists. And I have to admit, I was a little starstruck, especially the first time I met him. I had just started working for Komen, and I remember I was at the Brinker Award reception, and I completely starstruck. There is such a thing as scientific groupies. He made quite an impression. He is a leader in the breast cancer community, a supporter to Komen, and a driver for excellence in research every step of the way. I've enjoyed working with him so much, especially with his passion for the cure. He looks like a breast cancer researcher. Dr. Gray, I'm going to make you stand up. He looks like he's pink, right? You have to come up and stand up here anyway. So he looks like a breast cancer researcher, right? Pink tie, he's ready to go. But he actually trained as a physicist and an engineer. He's the perfect example of how we can apply some of this expertise from other fields and bring it to the difficult questions in breast cancer research. He's never going to forgive me. Now I've made an impression, right? He truly is. He has columns of honors and titles and awards behind his name, and I'm not going to get into them. But his expertise and knowledge, especially on metastatic breast cancer, is truly amazing. He's one of the best and the brightest. We are thrilled to have him as a Komen Scholar and thrilled to hear from him today. Thank you so much, and please welcome me in joining Dr. Joe Gray. I'm going to shake your hand. Thank you so much.
All right, well, Krissa, thank you very much. That was a very generous introduction, and it's a true pleasure to be here. I think one of the comments that was made earlier was that metastatic breast cancer is a complex disease to think about curing. I think that it is probably the most complicated problem that humankind is actually undertaking right now. I can think of nothing that is more medically and scientifically challenging than metastatic cancer. So what I'm going to talk to you today about is a new program for us. It is called SMMART. I hope at the end of this talk you'll remember what SMMART means, which stands for Serial Measurements of Molecular and Architectural Responses to Treatment. We worked really hard to find something that spelled smart. Anyway, the goal of this program is actually, it's a clinical program and it's a research program combined, and the goal is, in fact, to help us move aggressively in the direction of achieving durable and tolerable control of the disease. I have to disclose several conflicts of interest. For those of you who read the New York Times, you will appreciate that many of my friends have shown up on the front pages of the New York Times as having failed to disclose their conflicts of interest. So here I am. I have licensed technologies in a variety of directions. I hold ownership positions in a couple of small pharmaceutical and biotech companies. And most importantly, in the context of this presentation, I am going to talk about off-label drug use, and I have stock in a couple of drug companies. So be aware of that. So I'm going to come back to what it is that we're trying to achieve, and it is durable and tolerable control of metastatic cancer. And this is as I said, a research and a clinical talk. And I want you to appreciate the fact up front, since many of you are, in fact, most of you are dealing with this disease, is that this is an out-of-the-box talk. There is what I would call standard of care and conventional medical wisdom, and there is the SMMART program, and there's some overlap between them, but there's not a lot of overlap between them. And so I want you to appreciate the fact that the approaches that I'm going to talk about here really are experimental and should be thought about in consultation with your physicians, because what I'm going to talk about is not at all established standard of care. The other thing I want you to know is, since I have a background in physics, I am not a physician. What I am is somebody who's been working on breast cancer for about 40 years now, both from a research point of view and increasingly going toward the clinic. But most importantly, what I am is the husband of a wonderful person who is currently struggling with metastatic breast cancer. This focuses the mind. And so one of the things that I have increasingly focused on in the last few years is what to do. What can we do to bring better control of this disease forward? So as I thought about it, and as our colleagues have thought about it, there are really two ways to think about attacking cancer. One of them is broad. Let's study a lot of cancers and see if we can find a subpopulation of them that will actually respond to one of our emerging new therapeutics. And that is really the essence of precision medicine. Can we find some subpopulation of breast cancers that will respond to one of our new drugs? The survival curves, and I'm not going to explain survival curves since I suspect you all know about them as much as I do. This is the clinical trial that led to the approval of a CDK4/6 inhibitor. And what you can see in this clinical trial is that indeed we were able to find a subpopulation of patients whose experienced substantially longer progression-free survival on the drug, palbociclib, than those that were not on the drug. Very exciting, very important. But if you look at the survival curve, one of the things is you see a bunch of steps down. Every one of those steps down is the result of a patient being told, your tumor is no longer being controlled by this wonderful new drug. And so I think there are really two questions that arise at this point. Why did my tumor progress, and what are you going to do about it? That's the fundamental issue. And that's not going to be answered by a standard clinical trial. So what we're trying to do is answer that question. So we focus on understanding and durably controlling the individual cancers, and we do this by taking a very comprehensive, deep, and continuous view of the individual cancer. And so that's what SMART is about. Now, this is the problem that we're trying to address. This is the control problem. This is a patient with metastatic cancer. And one of the things that you will appreciate in the worst cases is that you're not trying to treat a single disease. You're trying to treat and control a collection of similar diseases, but that reside in different parts of the body. And so our take on this is we've got a lot of effective therapeutic agents that have been developed by the precision medicine community. The question that we're trying to deal with is which ones should we use, when should we give them, at what dose should we give them, and when should we change? This is the control problem. This is, Krissa mentioned that I have an engineering background. This is sort of the engineering control view of how one thinks about managing a cancer. So what we've tried to do is to put into place a program that will answer those questions. Now, when you do this, one of the things that Devon pointed out is that cancer is clearly a disease of the genes, and that is true, no question about it. But it's more than that. It turns out that how a gene in a cancer cell functions, how a mutation in a cancer cell functions, actually depends on where that cancer cell is in the body. And some of the drugs that we've got actually are stimulatory agents if given to cancer cells in the wrong environment. We need to know that. So what this brings us to is the realization that when we start thinking about trying to control these complicated cancers, we're not only trying to control the cancer cells, but we're also trying to, in essence, control the environments in which they live. So you've got the cancer cells, you've got the mutations in the cancer cells, and you've got then the immune cells that surround the cancer cells, the other normal cells. You've even got signals coming in from the brain that tell the cancers how to behave. And so what we're really trying to do is to manage this entire ecosystem. So we need to understand it. And in order to understand it, you could use model systems for sure, but our approach is to actually study this in the individual patient. And so the SMART program looks like this. This is a clinical program where a patient with metastatic disease enters. We acquire anatomic images of the diverse lesions throughout the body using MRI, PET, and CT. We then biopsy the tumor and run a deep series of analytics that I'm going to tell you about momentarily to try to understand what the vulnerable features are of that particular cancer. We then design a treatment based on that information. We start it, and then we monitor frequently. And I'll tell you a bit about that in a minute as well. But as long as things are going well, we continue. But if the cancer starts to progress, then we reanalyze, redesign the treatment, and start over. And we just keep going through this cyclic process. And so one of the things that this program is designed to allow us to learn is what are the resistance mechanisms that the cancers are using to escape therapeutic control. One of the things that I think I will fault most clinical trials on is we don't learn why we fail. We do a lot of clinical trials, we spend these billions of dollars, but we're not learning why we fail. And if you're going to fix it, you have to learn why you fail. And so one of the things that we're trying to do in this program is to learn that. So having done that, we also are trying to identify new therapeutic vulnerabilities that we can attack. But in order to do this, and in order to take advantage of that information, we have to run all of our analytics more or less in real time. Where for us, real time means 4 weeks. So from the time that we get a biopsy to the time that we have interpreted that biopsy and are prepared to make a clinical decision on it, it's 4 weeks. And that allows us to use the information that we acquire from our biopsies for the individual. Now, while we're doing all of this, we're also paying close attention to toxicity. And so the goal is not only control, but tolerable control. So, what are we trying to measure in order to identify these resistance mechanisms and therapeutic vulnerabilities? And the answer is all of the above. We have tried to put together a suite of measurement technologies that allow us to study cancer at many scales. So Devon introduced sort of the DNA, RNA, and protein level of a cancer. But if you think about what the proteins do, they organize themselves into molecular machines, and the molecular machines organize themselves into organelles, and the cells they organize into cells, and cells into tissues, and tissues into organs, and so on. And it turns out that the scale, this is about 10 orders of magnitude in scale size. And so on the one hand, you got features that are the size of a human head, and on the other end of the scale, you have features that we're trying to study that are 1 million times smaller than a human hair. So it's actually quite a daunting range of scale sizes that we're trying to deal with to discover mechanisms that the cells are using to escape and to discover new therapeutic vulnerabilities. And so one of the things over the last decade in Portland that we've been doing is putting together the suite of analytical tools that we can use to make all of these measurements. I remember in the completion of the first draft of the Human Genome Program, Bill Clinton was president at the time, and he stated publicly, because of the completion of the Human Genome Program, our children's children will know cancer only as a sign of the zodiac. He was a little off the mark on that one. It is way more complicated than that, but we continue to make progress. So, all right, these are the suite of tools that we bring to bear. And one of the things that we do is we try to bring these to bear on lesions, on individual cancer lesions that we know something about. And the material for that is acquired by our diagnostic radiology colleagues using ultrasound-guided biopsy after we've paid close attention to the lesion that we're trying to biopsy, and I'll come back to that momentarily. But once we've got these biopsies, we have a team of individuals that assemble in the diagnostic radiology suite, and they receive the samples from the diagnostic radiologist, and they prepare them in a variety of ways. They freeze them, they fix them in formalin, they prepare them for cell culture, they prepare them for electron microscopy, and then all of these samples are sent out to the various clinical and research laboratories for analysis. I'm not going to walk you through all of the different analytics that we're acquiring, but it is formidable. And one of the things that's really important if we're going to understand what's going on in cancer is that the clinical samples be preserved as well as we possibly can so that they don't change between the time you biopsy them and the time you do the analysis. And so in this workflow, the time from biopsy to the start of preservation is about 2 minutes. This has taken about 2 years logistically to figure out how to do that, but that's now in place. So, as I mentioned earlier, most of the research and the clinical assays are completed within 4 weeks, and it turns out that we are running a lot of samples. They're going in a lot of different laboratories, and we're generating a lot of information. And so we've also spent an awful lot of time trying to figure out how to put together computational systems that that keep us from getting things mixed up, and that allow us to manage the about 20 terabytes of data that we generate per patient. Now, 20 terabytes is an interesting number. It turns out that's about the amount of information that's encoded in the entire Library of Congress. So I'm now referring to this as about 2 LCUs, Library of Congress units. That's how much information that we are generating for these patients. And so you might guess that we don't memorize it. It takes quite a computational team to manage the information and to interpret it in ways that humans can consume of. Now, that's a real challenge, and I think it's probably the central challenge of the field, is how to go from really complex measurements into something that provides clinical or biological insight. But in any case, we've also worked fairly hard at developing user interfaces that allow us to take all of this distilled information and present it to tumor boards in a way that they can use to help make decisions about the management of the patient. And so we actually run 2 tumor boards at this point. We run a research tumor board, which is mostly populated by biologists and clinicians and whoever wants to sit in on these things. We run 1 a week. Each patient will be brought to the research tumor board, and we consider all of the information that we've got. Sometimes it may take a few hundred hours of interpretation to get through all of these data, but it then gets distilled down, and concepts are then presented to the clinical tumor boards for actual decision making. And I will say, this is the part where I have to point out that we are considering the use of drug combinations that are driven by the biology, and in many cases this takes us in the direction of off-label drug use. And in some cases it suggests the combinations of off-label drugs that have never been used before together. This is the risk part of this, and I don't want this to be underestimated. This is not something that is commonly done. Our IRB approval for this probably weighs about 6 pounds. So this was not a trivial thing to convince people could be done safely, but we are underway. So what I want to do now is, for the rest of this talk, is to just take you through a case study to show you what this looks like. Because the point I'm going to make at the end of this is that we've just started, but so far this is looking quite promising. And so I want to show you one study. This is a ER-positive breast cancer. Thank you, Devon, for introducing the subtypes. This was ER-positive, PR-negative, HER2-negative, initially treated with surgery, radiation, and adjuvant chemotherapy. This tumor upon diagnosis, was 7 millimeters in diameter, and yet it recurred. This widely metastatic cancer. It recurred on exemestane, on an aromatase inhibitor. And so we did the first biopsy, as I told you about before. It was still ER positive, PR negative, HER2 negative, but now we discovered a number of mutations and genomic changes that suggested therapeutic opportunities. And you can look at some of those. It had amplified this gene that we think would make it susceptible to a palbociclib. Still had wild-type estrogen receptors, so we thought fulvestrant would be good. And the omic analysis and some cell line studies that we did suggested that the tumor should be susceptible to treatment with the drug everolimus, which attacks one of the regulatory pathways that tumors use to stimulate growth. And so that was the starting point in this journey. So one of the things that we do when we monitor how well we're doing in these treatment responses is we look at the tumors using anatomic imaging, typically CT imaging, but we don't use the standard clinical descriptors of response and resistance. We actually quantify the size of every lesion in the body that we can see. And we keep track of the response of every lesion. And so if you see even one lesion beginning to progress, we consider that to be the start of escape from therapeutic control, and we want to do something about it. So you can see in, excuse me, in this plot that we've got, these are a few of the maybe 20 lesions that we kept track of in this particular patient, some from the spleen, some from the liver, some from the lymph nodes, and so on. And initially, this combination of drugs was mostly effective. But then after a few months, what you can see here is the beginnings of escape. Some of the lesions are beginning to grow. By the way, I will just note parenthetically, unlike most clinical trials, we keep track of every drug at every dose and know precisely to within the hour when the drugs were given, at least to the extent that the patients tell us that when they take the drugs is right. But nonetheless, what this allows us to do is to take a very close look at how the patients are responding to the drugs. The other thing that we do, and that we've really just learned in the last little while, is that if one frequently measures cancer protein biomarkers in the blood, they also very quickly and I think accurately report out on cancer progression. So these are cancer proteins in the blood, and you can see again that they suggest that we had early control with the drug cocktail that we used, and they also showed the onset of progression. Now, if we weren't at the time, but if we had been looking at this data closely, we would probably have known that this cancer was beginning to progress maybe 3 to 4 months before it showed up by anatomic imaging. So this gives us a tool to think about very sensitive control of the cancer. So once we recognized that we had a progressing lesion, then we went back, as I said, and rebiopsied the tumor. And reran the suite of analytics and asked what changed to make that lesion resistant to cancer or to a treatment. And so a number of things showed up. We certainly analyzed the DNA in the new lesion. And one of the things that we found was that there were some lesions, some mutations that were present in both of the metastatic lesions that we biopsied. Some were present only in MET1 and some were present only in MET2. And so what this allowed us to do was to recognize that we had sort of an evolutionary tree going on and that MET1 and MET2 were not the same. And in particular, MET2 had a mutation in this gene called PIK3CA, which is only important because that was a gene that was activating one of the pathways that we were attacking therapeutically with the drug everolimus. We also reran our cell line analyses and asked what did they predict, and they also showed that this tumor was now unlikely to be responding to everolimus. So suggested that the tumor had in fact escaped control from that particular therapeutic attack. But we, in essence, our computational team, in a few hundred hours of work, analyzed all of the omic information that we had generated on this patient and learned a number of things. One of them is that the tumor was probably escaping control with this palbociclib, the CDK4/6 inhibitor, because it had dramatically upregulated, amplified the cyclin D1 gene in that pathway. It escaped this everolimus because of the mutation I told you about, and because the tumor had upregulated this gene called BCL2, which is a part of a protein complex that cells normally use to self-destruct. BCL2 is actually a gene that keeps cells from dying when they should. And so this tumor had upregulated that. And the other interesting thing was that the tumor had now upregulated the androgen receptor. Not the estrogen receptor, not PR, not ER, but the androgen receptor. So that's interesting. We'll come back to that. All right. The other thing that the other set of analytics that we ran on this were designed to help us understand the environment in which the cancer cells live. And one of the exciting technologies that's been developed in the last few years in a number of laboratories around the country, including ours, is this ability to take standard tissue sections that the pathologists prepare in formalin and analyze them for multiple proteins. There are a variety of ways to do this, but basically, typically we will take affinity reagents for a particular protein, let's say the androgen receptor, and we can stain for that. Well, that's what's done all the time in histopathology laboratories around the world. What we've learned how to do now is to make that measurement and then erase it. Take a different protein, make that measurement, and first acquire an image of the first one, erase, acquire an image of the second one, erase, and so on. And so in any particular tissue section, we can now analyze for somewhere between 20 and 50 different proteins. And so you can see how they interact together. And what this helps you to do is to not only understand what the molecular state of the cancer is, but it allows you to understand things, for example, about the immune composition and whether or not the immune cells are there. And if they are there, are they in a state that would allow them to be effective against the tumor? There's clearly the natural killer cells, there's the T cells, there's the whole immune repertoire that's out there that we can now begin to interrogate to get a rather comprehensive view of the immune status of the cancer. So, one of the things, and I'm not going to walk you through all of the immune biology here, that would probably take another 2 hours. But the bottom line here is that this particular analysis, in fact, did indicate the potential utility of using these so-called PD-L1 inhibitors. These are inhibitors that are designed to reactivate the immune system. So we got a therapeutic clue from that particular analysis. Now, the other kind of analysis that we did is a little Star Warsy. So this now is in the electron microscopy world, and one of the electron microscopes that we have is sort of a bread slicer. What it allows you to do is to take the tumor and you scan the surface of the tumor, and then you use a second beam, literally, and you slice off 5 nanometers of the surface, and you scan it again, and you slice, and you scan, and you slice, and you scan. And that takes about a day, and it's fully automated, and then you take a few weeks of AI learning to render the data. But once you do render the data, this now begins to give you a great deal of information about the molecular anatomy of individual cells in the cancer. Now, I won't walk you through all of this. The blue thing is the nucleus of the cell. The purple things are the mitochondria, the energy factory of the cell. But the thing that we focused on here were these so-called macropenosomes, which is, in essence, a survival mechanism that the cells use to survive in nutrient-poor environments. And in fact, those little yellow bodies that are displayed here are the residual of leftovers, or I guess the mid-process of a process whereby a cancer cell will engulf its neighbor, a dying neighbor, and eat it. And it then reprocesses its neighbor's material to help it survive. So that's a strong survival mechanism. It looked like this particular tumor was using it. And that suggested to us the idea, well, if the tumor cells are taking up pieces of their neighbors, why don't we feed it drugs that are conjugated to proteins? It should convert the survival mechanism into a therapeutic vulnerability. So that suggested the use of a drug, for example, Doxil, doxorubicin encapsulated in a little lipid ball. So we did this. So the patient received Doxil and received the PD-L1 inhibitor. And these are now a continuation of the protein biomarkers in the blood. And one of the things you can see here is that we were very quickly, with this cocktail, able to regain control, in our estimation, of this particular tumor. One of the things that was really remarkable to us is that the evidence for establishing control was about 2 or 3 weeks. So there was a very rapid response, but not all responses you expect to be that rapid. But when you get a rapid response like this, you know you're on the right track. So it takes some of the guesswork out of how it is that we think about managing the cancer. Now you will see, as the time went on, we began again to lose control, we think, of the cancer. So possible progression. But we had some other ideas. We didn't have to go back and re-biopsy at this point. We still had some leftover ideas about what to do about this. The anatomic imaging showed the same thing. But if you go back to this analysis that I showed you earlier, one of the things that the analysis showed was that all of the changes that the second lesion had undergone certainly dramatically increased cell cycle progression, suggesting the possible use of a cell cycle targeted agent. And I talked about the upregulation of the androgen receptor, suggesting the possible use of enzalutamide. So that was administered, enzalutamide and capecitabine, and again, you can see that we have reestablished control. Now, the other thing that's apparent on this is a little break in the rate at which the protein biomarkers are going down. The reason that break is there is because we did a dose de-escalation of capecitabine to minimize toxicity or to reduce toxicity. And what these data show you is, well, it did indeed reduce the rate at which we were diminishing the tumor, but the tumor is still diminishing. So we feel that this shows us that we've got control of the tumor still, and so we're quite confident to be working at this lower dose. So that's the general kinds of control strategies that we are embarked on. Now, there's a lot more left to learn about these tumors. So what else is out there? Assuming that we lose control again, what are we going to do next? And so we're still thinking about that. One of the things that is on the plate is this upregulation of BCL2. We know it's upregulated. We haven't attacked it yet. Venetoclax is a perfectly good drug to think about that, and so that is on the plate for possible future use. I want to come back to the immune system. So this is one of the multicolor images that we, maybe this movie will run, or maybe not. Okay, well, one of the things that we've done is to go back and reinterrogate the immune system. And one of the interesting things that came out of this is that these are mostly cancer cells that are present in this image. If you look closely at them, and it would have been better if the movie ran, but anyway, you'll see that they're sort of organized into clusters. And if you stain for immune cells, one of the things that you will appreciate is that the immune cells are actually not in those clusters. They're excluded from the clusters. So why is that? And so one of the other devices that we have is this so-called, in this case, serial block space imaging scanning electron microscope. Kind of like the other one. You scan the surface, but now you have a diamond knife that goes in and slices off the surface. Its advantage is that it allows you to see a little bit more real estate. But we wanted to understand why those tumor clusters actually existed. So this is sort of a scan through the electron microscopes now, and when we come back up, I'll render out the different kinds of cells. So the greens and the blues are fibroblasts, and the reds are the cancer cells. And one of the things that you will appreciate from this image that you really didn't get from standard histochemistry is the extent to which the tumor cells appear to actually be recruiting the fibroblasts to grow up and around them. And to secrete the extracellular matrix that we think is forming a barrier to the escape of cancer proteins to the immune cell, and we think that this is the barrier that is in fact keeping some of the immune cells from making direct contact with the cancer cells. Hypothesis? Sure. But it certainly suggests the idea of attacking the fibroblasts with MEK inhibitors to open them up. So a new therapeutic approach that's aimed not at the tumor cells directly, although using agents that were designed to attack tumor cells, but rather attacking the microenvironments in which the tumor cells sit. Now, if you drill down and ask the question, how do cancer cells interact? This is brand new data. This blows my mind. You cannot see any of this in the light microscope, but each of those colors is a different cancer cell. The little protrusions that you're seeing sticking out there are about 1/100th the diameter of a human hair. You can't see them in the light microscope. But what's very clear is that most cancer cells, at least the ER positives and the HER2 positives, manifest these things in great abundance. Didn't know they were there, but now that we know that they're there, one of the things that we've learned is that they're quite dynamic. So this is a different kind of microscopy, but it allows you to see not only the protrusions, but the proteins that are located on them. So this is the location of HER2, one of the receptors that we talked about before. And what's interesting is that these protrusions are decorated with HER2. They're also dynamic. So if you feed it with a growth factor, one of the things you'll see is that these little protrusions have reached out, grabbed a hold of the part of the environment where they reached to, and they literally pull the cell back, pull the cell to them when the growth factor is given. So this is a way that cells can sense their environments, find something that's interesting, and move to it. This is a roam through a different part of the cancer. The blue thing is an immune cell. The other cells are tumor cells that are showing the protrusions. You can almost see the cancer cells reaching for the cytokines that the immune cells are elaborating. So, who cares? Well, basically, I care because one of the things that we are going to try to do is to cut off the protrusions. We actually know a lot about what drives them. And so this is a way of thinking about chopping off the protrusions to keep the cells from moving and to increase the efficacy of the drugs that we've already got. So I'm sort of getting to the end of the talk here, but basically to summarize, this SMART program does allow us to discover the resistance mechanisms and novel therapeutic opportunities as they arise during the course of treatment. I think that the analytics that we're bringing to bear give us unprecedented analytical depth to understand what's really going on, and there's a lot to discover. And we do this in sufficient depth that we really come up with a treatment for every patient. So far, there's been nobody that's come through the program, N of 10, for whom we haven't come up with 2 or 3 different therapeutic opportunities. We think that the rapid or the frequent response measurements that we're making allow us to identify when we've got ineffective drug treatments and to allow these things to be discontinued early so we can switch to something that's going to be more effective. We can do tox monitoring and dose de-escalation to make the treatments more tolerable, and we're providing detailed information to guide our tumor boards. I made this point earlier, but we have a lot to choose from in this enterprise, and outcomes so far, we've put about 10 patients on it. I would say that we had 2 non-evaluable patients that basically failed before we could get drugs to them, but the rest of them are all still under control. So that is exciting, very early days, but we are continuing to push the program and currently enrolling in 3 trials. Zahi Mitri is the lead oncologist on this, and if anybody wants actual medical information about this, you should contact Zahi and not me. You know, the bottom line here is that we can execute it. We think we can execute it safely and in real time. We learn from every patient, and we use the information for that patient. We're doing it now. Drugs are off-label and in new combinations, and I will say that insurance does not cover that. This is being covered solely by philanthropy. Otherwise, we couldn't do it. As I say, a treatment for every patient, but with uncertain risk. And I want to end with the uncertain risk because this is still early days. But the most important part of all of this is the last slide. And that is, this is a real serious team opportunity. We have the operations team that makes it work, the financial team that manages the money, the clinicians that carry out the studies, the diagnostic radiologists that get the biopsy teams that manage the samples, Search Path that helps get the samples. We have the night diagnostic laboratory who are running all of the clinical diagnostics. The pharmacy team tells us about toxicity. The bioinformatics team helps us to manage the information and interpret it. The biolibrary team keeps track of the samples. And then we have a whole cast of biologists and clinicians and so on that work together to try to interpret all of this information to give us clinical insights. We have protocol teams that help us to carry out the clinical trials, and we have people that help us make contact with the private sector who are attempting to get us the drugs that we're using. We have great support from our patient advocates and fundraisers. Thank you.
Thank you very much. I'm going to have nightmares tonight. Are you? I mean, these little things coming out of the tumor cells and no, what's going on in my body? I don't know. And you have 9 people on it, and that's 3 million people who are part of this, right?
Yes. Well.
Okay, so this is an expensive program, but it should get us, some really, really good data. And the cool thing about it is that it's really expensive and really laborious now, but when we get better technologies and when we get better ways of doing things and quicker ways of doing things, it's going to be just like the Genome Project that was billions of dollars, several years ago, and maybe $100 today. So there's a lot of hope in areas like this. But on behalf of our program, hopefully I'll give you the right one. Yes, I do. You are in Orange County. So we did give you some oranges, and because they don't have those in Oregon. And we thank you so much for your time and all the things that you do. Thank you so much.